This trial arm within the ESMART platform trial is assessing the combination of a PARP inhibitor, Olaparib, and an ATR inhibitor, ceralasertib. And the aim was of this first part of this study to find a dose which is well tolerated in children and to assess preliminary activity. Given that we have molecular profiles at relapse from the patients, we are in a position to trace back and to identify molecular biomarkers for response. The background why we wanted to assess this combination is that in pediatric cancers, responses to single agent PARP inhibitors are not really expected. This is in contrast to adult cancers, where a lot of patients in specific tumor types like breast cancer, ovarian cancer, prostate cancer, pancreatic cancer, do have alterations in genes called BRCA1 and 2, and a few other ones, which are really driving this disease, and where the effect of the PARP inhibitor is to kill these cells. And these patients really benefit from single agent PARP inhibitor treatment. In pediatrics, the situation is very different. We don't have these mutations, but we still have evidence from work in the laboratory that PARP inhibitors might sensitize these tumors and that they have a role to play, but that these tumors have kind of a primary resistance to these PARP inhibitors, and because they overactivate another pathway which is driven by a key protein or kinase which is called ATR. So assessing this combination of ATR and PARP inhibitor, we were thinking could overcome this primary resistance and really be of benefit to these children with relapsed refractory tumors. The study design within this ESMART platform study, all trial arms are assessed separately, and they have a two-stage design. The first stage is a phase 1 dose escalation, which uses a Bayesian design. In this case, it was a Boyne design, which is meant to identify a safe dose to move forward in the phase 2 part, which is the assessment of the preliminary activity of the combination. And in this specific combination of the ATR and the PARP inhibitor, it's a Simon two-stage design, which moves on to the second stage if a specific number of responses has been observed in the first stage. And we are looking at 2 cohorts in this second phase, and these cohorts are tumors with deficiency in homologous recombination in the broad way and sense, looking at our molecular profiling data. And the second cohort is tumors which have high replication or transcription stress based on proven presence of, for example, aberrant transcription factors like EWS, FLY1, or PAX3, FOXO1. At AACR this year, we've reported the phase 1 part of this study, which was the dose finding part. We've recruited 18 patients, and we've identified safe dose, which is comparable to the dose used in adult patients. And already within this limited number of patients and the dose finding, we have seen preliminary responses. So we had a partial response, which was confirmed in a patient with a pinealoblastoma and in a patient with a neuroblastoma. And we also had prolonged stable diseases in 3 more patients for several cycles, one of the patients being on study for more than a year and ongoing. These results are extremely important and also very exciting because they show us in this first-in-child study that we can combine safely a PARP with an ATR inhibitor and that we indeed can see responses. We have proven a safe dose for the older patient cohort with 12 years and onwards. We don't yet have enough patients of younger age to declare the safe dose for the younger patients. So work in this direction is ongoing, and we are also awaiting the results of the pharmacokinetic analysis of both drugs to see if the dosing schedule is the right one where we are and if we can further optimize it. Most importantly, we have the opportunity to go now back to our tissue blocks of the patients and the molecular information, which is already available from sequencing from the tumor samples at relapse. And by going back to the laboratory and to the bioinformatic colleagues, we are able to see which alterations in the tumor can really be linked with the responses or the rapid progressions in the clinic, to identify future markers which can help us to choose the right patients for this combination and to avoid treating patients who very unlikely will benefit from this combination. I would like to thank for the opportunity to speak to you and share the data. And I do think that for pediatric oncology, the approach to have comprehensive molecular sequencing available and doing an early phase clinical trial and then go back to the bench and to the thinking process and refine selection criteria for a trial is key going forward for us to drive forward pediatric oncology drug development. And another opportunity out of this is that if we have hints which could be the right biomarkers, we also can go back in the laboratory and do more research with this gained knowledge. And so it's an iterative process between preclinical work and clinical work. In this way, one day we hopefully are beating cancer in childhood.